Okay, welcome everyone. It is really my great pleasure to introduce my friend and collaborator, Alan Van Orden. Alan is a native of Idaho, and he was a grad student in Rich Sakeley's lab, the infamous Sakeley lab, a great one at Berkeley, where he studied silicon carbon clusters. And then he was a postdoc in Dick Keller's lab at Los Alamos and contributing, no doubt, some fantastic ideas and techniques in single molecule spectroscopy, which no doubt would have helped and contributed to a Nobel for Dick if they hadn't decided to give super res a Nobel nod, and that kind of negated any single molecule stuff is how everyone kind of looked at it. So that was a missed opportunity for all of our single molecule spectroscopists. Alan is a chemistry professor at Colorado State University. He's been a faculty fellow of Air Force Research Lab, of the Institute for Material Science, of Center for Integrated Nanotechnologies, and all kinds of great contributions and collaborations around the country. So with that, Alan, take it away.
All right. Thank you so much, Jen. It's really good to see you, and it's great to be here with you today. I am going to get my slideshow up and running here. And I'm going to pull up my laser pointer. So today I'm going to be telling you about some work that has been a collaboration between my lab at Colorado State and the Center for Integrated Nanotechnology at Los Alamos. And this is where I got to know Jen quite a few years ago. And we've been continuing to try to do things together ever since. And so I want to start out by acknowledging some of the coworkers that we have. This is a pandemic group photo. And it has people from CSU. So up here on the left is my colleague at CSU, Marty Gelfand, who's been working with me for many years. And then here on the right is a former grad student, Duncan Ryan, and he has been a postdoc at CINT for the last few years, and is now just got converted to a staff scientist position. So he's a staff scientist at Los Alamos. There's Yushan, and this is the connection with Jen, is that this is one of Jen's former postdocs that we did some collaborations with. And here's Megan Dunlap, who is my current graduate student, and she has also been spending quite a bit of her graduate career working at Los Alamos. And she is going to, much to my chagrin, is going to be graduating soon and landed a postdoc position at Pacific Northwest National Lab. So that's an exciting time. Peter Goodwin is our CINT collaborator at Los Alamos, who many of you know. Down here is John Gann. Farshad Abdullania was a former grad student who is now a postdoc at Scripps. And I just found out just the other day that he has been given a NIH postdoctoral fellowship. And then this is my newest grad student down here at the bottom, Courtney. And I also want to show some historic pictures of my group because I'm going to be telling you a little bit of history. And so some of the people who contributed to that history are in some of these older pictures. So up here on the left, that's a picture that's probably about, I don't know, 10 or 11, maybe 12 years ago. And this guy here in the middle, some of you know, is Doug Shepard. And Doug was one of my grad students, and he is kind of credited with getting us started along this trajectory that I'll be telling you about. He's currently a professor at Arizona State University in the physics department. Kevin Whitcomb is another grad student who worked with Doug and did some important contributions as well. And then down here is a more recent picture, and here you can see Megan and Farshad back when Farshad was still a grad student in my lab. And one of the things I noticed when I look at this picture is either I am getting taller or my students are getting shorter. And I think it's the latter. Okay, so um, let me start out just by telling you a little bit of basic information about single molecule detection. Okay, so I started in the single molecule detection field many, many years ago as a postdoc at Los Alamos, working with Dick Keller. And Jen mentioned Dick and he's one of the pioneers of single molecule detection. And some people argue about who was the first single molecule detection, and there's a case to be made that Dick Keller was really the first person to demonstrate single molecule detection under ambient conditions. Okay, and so that's an important thing, is being able to do things under ambient conditions. The Nobel Prize went to the person who first detected single molecules, but that was done under cryogenic conditions. And so people argue who is really the most important contribution, but that's an argument that will probably go on forever. Anyway, so what you're seeing up here on the left is some molecular structures of some dye molecules that are, it turns out not all molecules can be detected using single molecule fluorescence methods, and they have to have some very special properties. They have to have very high fluorescent quantum yield. That's this Phi number here. They have to have large absorption cross-section. They have to be relatively photostable. And there's only really a handful of molecules that can actually do this. And so I'm showing you a few examples here on the left. And basically what we're doing when we do single molecule detection is we're pumping the molecule into an excited electronic state, and then we're waiting for the molecule to decay back into the ground electronic state, and we're detecting the photon. So this K21 process that you see here, that's what we're detecting. And you can see that there are some competing processes going on as well. The molecule can cross into a triplet state, and when it does that, it becomes non-radiative for a period of time, and we have this property called blinking that we often see at the single molecule level, and that's one of the things that can give rise to blinking. But what we're really doing is we're looking for this K21 process, and we pump the molecule over and over again. And so this process is something that happens on the nanosecond time scale. So in principle, about every, we can see a photon every few nanoseconds if we have the right conditions. And that means that it's possible to detect 10 to the third to 10 to the sixth photons per molecule per second. And so that's the basis for single molecule detection. It's the fact that we can, the right type of molecule can undergo this excitation many, many times, and we can detect the photons that are coming from that molecule in a very rapid time frame. What we're going to be talking about today is a different type of single molecule, quote unquote, and that is going to be the semiconductor quantum dot, semiconductor nanoparticle. And so this picture is just showing us a piece of semiconductor material that we're going to shrink down in size, make it smaller and smaller and smaller, so that it starts to behave like a single molecule. And so as we make this semiconductor smaller and smaller and smaller, what happens is that these electronic bands that you see here, the conduction band and the valence band, go from continuum of energies to discrete energies. And then as we get down to the quantum, to the level of a nanoparticle, so this is down at around 2 to 10 nanometers in diameter, we get to this point where the conduction band and the valence band have very discrete electronic states that are similar to molecular electronic energy levels in molecules. And so the quantum dot is going to start behaving like a single molecule. And we can excite the quantum dot using light, and then when the excitation relaxes, we see photons, and we can do this in the same manner that we talked about with the single molecule. So here's a picture of an example of a type of quantum dot that we've studied. We have the zinc sulfide or the zinc selenide core surrounded by a zinc sulfide shell. And we have the valence band and the conduction band states. We have these little trap states that can sort of behave analogous to triplet states in molecules. And they can cause the quantum dot to go into non-radiative states. And so what happens is that as we monitor the emission coming from a single quantum dot, what we're seeing is that it's emissive for a period of time, and then it shuts off and it becomes non-emissive, and then it turns back on, and it goes through this on and off blinking process over and over again. And so that's an important property of the quantum dot emission, is this blinking. Another important property has to do with the lifetime. So we mentioned this idea that that single molecules and quantum dots, it takes a certain period of time for the fluorescent signal to occur, and that's referred to as the lifetime. And so one of the measurements we do is we take data like this, and we monitor the intensity, the number of counts per 10 milliseconds in this case, and then at the same time, we monitor the fluorescence lifetime. And this particular quantum dot has a fluorescence lifetime on the order of about 30 nanoseconds. And then we make what's called a fluorescent lifetime intensity distribution. And that shows the relationship between the lifetime and the intensity. So on average, we're seeing about 40 counts per second, and we're seeing about 25, 30 nanosecond lifetime for this particular quantum dot. Okay, so now I want to talk about what is it that we're actually trying to study. We're interested in looking at not just single quantum dots by themselves, but we want to look at small groupings of quantum dots. And why would we want to do that? And the answer has to do with a lot of us kind of think about this idea of emergent properties. And, emergent properties happen when you have individual entities that combine together and interact with each other, and they give rise to properties of the system, of the group, that are not found in the individual entities. And so in the case of these quantum dots, we're thinking that if we put them together in these small groupings, that they can interact with each other. And there's ways that they can do that. They can share, they can exchange energy, they can exchange charge carriers, and that those interactions are going to produce emergent properties. And so you see here that we have these little dimers, and we have trimers, and we have different shapes of trimers. We also have single particles. And so each one of these each one of these groupings of quantum dots is its own individual entity that is going to have its own emergent properties. And that's something that's probably, I would say, the big discovery that we've made so far, is that some of these little groupings of quantum dots do exhibit what we would describe as emergent properties, and others don't. And why is that? So the question, I think, is that's driving us forward. What are the rules that govern this idea of emergent properties? What are the structural features that give rise to that? And why do some of them have it and some of them don't? And so that's what we're going to be trying to talk about. Okay, this is an electron microscope image, by the way. It's not a fluorescent image. I will show you some fluorescent images. But this just kind of gives you an idea of the structures that we're looking at. All right. So I want to tell you about, this is kind of a historic slide. It shows an experimental setup that we developed many years ago with the idea of studying these types of systems. When we originally developed this setup, we were thinking of looking at, all kinds of different things, biomolecules and, cells, and we ended up focusing a lot of our efforts on quantum dots. And the idea is that it combines a single molecule fluorescence microscope with an atomic force microscope. And so what we do when we do single molecule microscopy, what we do is we take light and we focus it onto our sample, and we make a little optical detection region. So the little focus that's shown here is the optical detection region. That's where we're exciting our sample with the light, and then the emission that comes from the sample is going to be collected using this same lens system, this same microscope objective, and we'll use our avalanche photodiode detectors to detect that light. And so that's the basic idea. In these samples, the quantum dots and the clusters of quantum dots are immobilized on a surface, and we use a piezo stage to move them around and look for different particles and groupings of particles. And then we have an atomic force microscope that we use, that we co-align with this optical detection region. And so we then take a topographical image of that little light beam, and we co-align those two. We co-align the optical detection region with the AFM probe tip just by monitoring the light that's being scattered off the probe. And so we can then collect a high resolution topographical image that shows us what we're looking at inside of that optical detection region. And so we use this setup to get a lot of our preliminary information on these quantum dots. And this is Doug Shepard's experiment that he set up. Okay, so here's kind of a zoom in picture of the optical probe region. Here's some quantum dots and some little clusters of quantum dots that are immobilized on the surface. And there's our AFM tip that we're going to go in and we're going to probe just that little small optical probe region. Okay, and we're going to collect the fluorescence coming from the quantum dot. And so there's our quantum dot blinking that you see there. And then we're also going to take an image of that little probe region. And in this image, we see just one single isolated quantum dot. And then we move our stage around until we see a cluster of quantum dots. We see the fluorescence coming from that cluster. And this is a first example I'm going to show you of an emergent property. Okay, so this blinking behavior that you see for this cluster of quantum dots is very different from what you would see if you had multiple, like here we're showing that there are 4 quantum dots clustered together. If we had 4 quantum dots that were independent of each other and not interacting with each other, we would not see this type of blinking behavior. We would see just kind of an average fluorescence emission. But this very fast, rapid, intense blinking that you see is an emergent phenomenon that has to do with the interactions of those particles. Okay, and so we look at our AFM image, you can see a blob. And we can't really, one of the things that we found out fairly early on that was a little disappointing is that we couldn't really resolve the individual quantum dots with our AFM. And so we see the way, so what we can do is we can look at the volume and the brightness of that little blob, and we can tell that there's probably 3 or 4, maybe 5 quantum dots in there, but we can't really resolve the individual quantum dots. And you can compare that to the previous picture. That's what an individual quantum dot looks like. It's just a little blip. And then the blob, the bigger blob, is the cluster of quantum dots. Okay, so another measurement that we do is the time correlated single photon counting measurement. And so this is how we get the lifetime information. And I'm showing you again this picture of an energy level diagram. And what happens is that when we excite the particle, it stays in the excited state for a period of time. In the case of a single molecule, it's usually on the order of a few nanoseconds. In the case of a quantum dot, it's more like tens of nanoseconds. And so we try to measure that lifetime. And we do that using a pulse laser, and then we just monitor the elapsed time between the photons and the laser pulse. Okay, so here's some data that we get. We do a bunch of these measurements on single quantum dots and clusters of quantum dots. And then for each one, we do what's called an autocorrelation analysis. And that's telling us how fast the particles are blinking. So if we have an individual isolated quantum dot, the blinking behavior is prolonged and we get this broad autocorrelation function. If we have multiple quantum dots that are not interacting with each other, we get this same broad autocorrelation function. But if we see particles that are interacting, the blinking becomes very fast. And so we see this, this rapid decay in the autocorrelation function. And then we also measure the lifetime. And when we look at the lifetime, we see that for an individual isolated quantum dot, we see a single exponential decay in the lifetime that has a lifetime of about 20 to 40 nanoseconds. If we see a cluster of non-interacting quantum dots, we see this same single exponential decay behavior. But if we see a cluster of interacting quantum dots, then we see this fast component to the lifetime. So we see the faster lifetime, we see the faster blinking, and those are some of the emergent properties that we see for clusters of quantum dots. Okay. So another way that we can look at this is by taking a bunch of data that shows a single isolated quantum dot. You can see the fluorescent lifetime intensity distribution has a single intensity, a single lifetime. You can see the AFM image shows a single quantum dot. This is a picture that shows some data that we have for a group of quantum dots that are not clustered, that are not interacting with each other. And the interesting thing here is that you see the same, 20 to 30 nanosecond lifetime. You see a broad distribution of intensities. You see higher intensities, but you don't see any evidence for this rapid blinking, and you don't see the short lifetime component. Only when we have the cluster of quantum dots, and there's the AFM image of a cluster, we see the fast blinking, and we see this short lifetime component in the FLIT. So these are kind of our signatures that we have that tell us that these quantum dots are interacting with each other, and they have some emergent properties that are not present in the individual isolated quantum dots. All right, so we can come up with some models that sort of explain pretty well what's going on. And the model that we have that pretty well explains our data is this energy transfer model. And so the idea here is that we can have high intensity emission, and we can have low intensity emission. And the low intensity emission is really governed by this competition between the radiative decay and the energy transfer to a dark acceptor. Okay, and so the idea here is the acceptor is blinking on and off. And when the acceptor blinks off, the energy goes into the acceptor, and there's no light that comes out. And so it's basically the acceptor quantum dot is sort of governing the emission of the entire particle. All of the other particles are funneling their energy into this acceptor. When that acceptor blinks off, there's no more light that can get out. So it's kind of like an energy sink. And this kind of explains the change in lifetime that we're seeing. If we have non-interacting quantum dots, then we just see lifetime components that are going to be the same for each quantum dot, that are these longer lifetimes. If we see energy transfer between the quantum dots, then we see this short lifetime component. All right. Okay. I want to tell you very quickly about another emergent property that we have found for these quantum dot clusters, and that is that they undergo this phenomenon called anti-bunching. So let's just show some data here. This is what we're looking at here. This is another one of these correlation functions that I talked about earlier. And the idea here is that when we have a single emitter, so like a single molecule or a single quantum dot, that single emitter can only emit one photon at a time. And there is no possibility that we can see simultaneous photons coming from a single emitter. And so this little dip that you see here at time zero, at delay time zero, what we're doing here is we're trying to measure the number of coincident photons that we see. And if there's no signal, in this case, there's a dip, because there's no signal at time zero, that means that there are no coincident photons. And if there are no coincident photons, it means that we have a single emitter. Okay, so a quantum dot behaves as a single emitter. So now the question is, what about these clusters of quantum dots? Are they going to have show bunching or anti-bunching? Well, let's take a look at this big giant blob first. The big giant blob that you see here shows that there's a big large peak at this zero delay time. Okay, and that big large peak is showing that that we're detecting lots of coincident photons. Let's go and take a look at these two quantum dots that are separated from each other. And we are seeing a peak. It's a smaller peak, but we're seeing a peak at that zero delay time. And so that's telling us that we can have coincident photons if we have two independent quantum dots. Here is the single quantum dot again, no peak in the middle. And here is a quantum dot cluster. And that quantum dot cluster is showing that there is a little tiny peak. So there are a few coincident photons, but if we compare that peak to what we should be seeing if the quantum dots were non-interacting, it's much lower. And so we're seeing some anti-bunching associated with the quantum dot cluster. There's a little bit of probability of detecting multiple photons, but it's much smaller than what we would expect based on if they were just independent particles. Okay. All right. So, and then here's our model that explains how we can see anti-bunching in these particles. Okay. So all of this stuff these are things that we've kind of worked on. It's kind of like a historical introduction of what we've done. And like I say, Doug Shepard, Kevin Whitcomb were the important people who did a lot of this work back in the 2010 to 2015 time range. But it left us with a lot of unanswered questions. And the big question is, why, like I said before, why do some of the particles behave this way, give rise to these emergent properties? Not all of them do. We can show examples of different particles that don't have this. And so the question is, what is the structural basis? Does the arrangement of the particles matter? The number of interacting particles, how they're oriented, how they're shaped. What are the things what are the important features? And then the other thing we wanted to do is I'm going to go back to this picture that shows the model. An idea that we had when we looked at this model is that if we could spatially resolve the emission coming from these clusters, then sometimes we should see the emission coming from the acceptor, and sometimes we should see the emission coming from the donor. Okay, as the acceptor particle blinks on and off, we should see the emission. If we could spatially resolve it, we should see the emission coming from different places. And so this is where we became very interested in the 2014 Nobel Prize in Chemistry. Okay, this is what Jen was talking about, is some people thought that Dick Keller should have gotten this, but he didn't. But these are the people who got it. And, I'm not going to begrudge them their Nobel Prize, because this is a very useful thing that they did. They developed the super resolution imaging microscopy. And so we started to think about the idea that we could use this super resolution imaging microscopy to image these quantum dot clusters and to see where is the emission coming from in these clusters. Okay, so let's just take a little bit of just a few moments to explain a little bit about super resolution microscopy. So if we take a sample that has some quantum dots or some single molecules that are spread out on a surface and immobilized on a surface, we can use a wide field camera to image the fluorescence. So in our experiments, we tend to just look at one single point at a time. But another way of doing this is to illuminate a large region of the sample. and then use a wide field camera that shows fluorescence from multiple individual particles. So each one of these little dots is probably, an individual, the fluorescence emission from an individual particle. Now this fluorescence is going to be spread out over some pixels, and the size of the image is going to actually be much, much larger than the size of the molecule. So we're not actually measuring this, we're imaging the light that is coming from the molecule, but we're not imaging the molecule. The light coming from the molecule is spread out over a wide area. And so here we see the kind of the point spread function spread out on the individual pixels. And this is just from one of these little, we're zooming in on just one of these little dots here. Well, a point spread function is a light distribution coming from an individual point emitter that kind of looks like this. It has a intensity distribution that you're seeing here. We can approximate it as a Gaussian shape, which is the red curve that you see here. And what you notice is that the maximum point of that point spread function matches up with the position of the emitter. And so if we can analyze this data and find out and get a good estimate of what does the point spread function look like, we can find the centroid position of that point spread function, and that is the location of the emitter to within, it's possible to get that down within a few nanometers. So what that's called, if we do that on a single molecule, it's called single molecule localization microscopy. What the thing that makes it super resolution imaging is that if we have multiple emitters, so here's a picture that shows some images, and what's happening is that this image here actually has two emitters that are contributing to that image. This image right here has three emitters that are contributing. And what's happening here is that these emitters are not on at the same time. They can blink on and off. And so sometimes, or they can bleach. So sometimes one of them is on and the other one is off. And so we can do this super resolution, this single molecule localization microscopy. If we monitor that as a function of time, we can find points in time or where only one molecule is present, and then we can locate that molecule. That molecule blinks off, the other molecule blinks on, we can locate that molecule. And so over time, we can build up a super resolution image on the right that shows us what is actually happening within that image there. Okay, so that's the idea of super resolution microscopy. And people use acronyms like PALM and STORM and things like that have different meaning, but it's basically the similar idea. So we wanted to do this super resolution microscopy, but we also wanted to do to spatially correlate this with electron microscopy, because we want to get an idea of what the structures look like. And so we put our quantum dot clusters on a TEM grid, and then we position them above the microscope. And then here we're seeing the point spread function for a grouping of quantum dots. And so we're going to monitor that point spread function. And the idea is that if these quantum dots are blinking on and off, then as we monitor that point spread function, we can see it shift back and forth as the different quantum dots are blinking on and off, and we can eventually build up a picture of those quantum dots. And then we take our sample and we bring it over to an electron microscope, and we image those particles to see what we have. Okay, so here's some data. First piece of data is a single isolated quantum dot. Here you can see this is basically the super resolved image. So what we're doing here is we're monitoring the emission onto a camera, and we're collecting the point spread function. And then about every 100 milliseconds, we're analyzing the data that we get and finding out where the centroid is. And then we're graphing out those centroid positions. Each one of these dots represents a different measurement of the centroid position for each 100 millisecond worth of data. And then we color code it according to intensity. And so if we only look at the really highly intense features in the green, they're really bunched together with a you know, we've got about a 5 to 10 nanometer distribution. And then the lower intensities have broader distributions. But the key here is that we're able to see we're able to kind of hone in on where that particle is. And then we can compare that to our corresponding electron microscope image, and we can see what we're looking at. Okay, let's do the same thing for a pair of quantum dots. And this is an interesting picture. And here we're seeing that our point spread function is spread out, kind of consistent with the size of the particles, but we're not really seeing any, we're not really resolving the independent particles. And the reason we're not is because both of these particles are emitting at an equal rate. And so most of the time when we see the most intense emission, both of them are emitting at the same time. And so our most intense emission is coming from the center, the average position of that cluster. So this is an example of two quantum dots that do not have emergent properties. They're just behaving like two independent quantum dots. All right, and then here's an interesting one. This one shows that most of the emission, this one shows that there's a hot spot, and the hot spot is the top left, or the one here at the top right. And so we call this an emission hotspot, and it's showing us that most of the emission is coming from this top right one. We think it's because the energy is being funneled from a donor to an acceptor. Okay, and we can show some more pictures that have, there's a trimer and there's a hotspot that goes with this one. There's another trimer and there's a hotspot that's in between the two up here at the top. And so we see a key idea here is that we're seeing different behaviors, and sometimes we see evidence for emergent properties for energy transfer, and sometimes we don't. Okay. Okay, so that brings us to the thing that we've been working on for the last few years. So let me point out, okay, here we go. All right, so we're trying to explain why do we have these emission hotspots? Okay, it could be because there's energy transfer from donors to acceptors, and that most of the emission is coming from the acceptors. Or it could simply be that we just have bright and dark quantum dots, and that there's not really anything interesting going on at all. We could have dark quantum dots that don't emit. And so we still have questions. You know, after taking all of this data, there are still questions. And so what we wanted to do is recall that this signature for energy transfer that we've talked about earlier has to do with the lifetime. It's really the lifetime and the intensity combined. Those things combined are what we really need to be able to characterize the electronic interactions. And so what we've been doing ever since 2017, and Jen remembers very well when I came to Los Alamos in the summer of 2017, my goal was to try to develop a new time-resolved super resolution microscopy setup. And so we've been doing that for several years now. And the idea here is to do this same super resolution imaging that I've just talked about, but to have lifetime imaging, to be able to tell what the lifetimes are of the different particles. Okay, so here is our setup that we came up with. We are using, and so basically, let me point out the problem here, is that most of the super resolution imaging microscopy is done using CCD cameras. And those are very nice cameras. They're very sensitive, but they don't have timing resolution. If we want to measure lifetimes, we have to have nanosecond time resolution. Okay. And so how did we get our nanosecond time resolution? And the way we did it is we replaced our CCD camera with a bundle of optical fibers. Okay. And so we found these square fibers and we formed them into a bundle. And then what we're doing is we're using the apertures of those square fibers as a little camera. And we're just using a little 2x2 camera that works just fine. And then we're sending the light from those optical fibers onto our favorite type of detector, which is an avalanche photodiode detector. Okay, if you come from Dick Keller's version of single molecule detection, then your favorite detector is an avalanche photodiode detector. So using this fiber bundle instead of a CCD camera allows us to use our favorite detectors. And with those detectors, we can monitor the timing of each detected photon relative to the laser pulse, and we can find the lifetime of the quantum dots. Okay, so this is just showing us that we're going to use these. We just need 4 pixels to predict the point spread function, and then we can find the centroid position of that point spread function, and it gives us the position of the particle. Okay, so here's some data that, again, starting with just one single isolated quantum dot, we are seeing our intensity trace, and we're seeing that it blinks on and off. We're seeing our lifetime trace, okay, because we're able to not only monitor the total amount of signal, but we're able to monitor the lifetime associated with that signal. And we're also able to measure the X and Y position. And so that's what, using the 4 pixels and finding the point spread function, we find the maximum of that point spread function. That gives us the X and Y position. I'm just showing the X position here, but we also have a corresponding graph for the Y position. And so we can get the same image that we got for the in the previous example. This is just the intensity, or this is just the each one of these dots is just the point spread, the center of the point spread function that we found for each 100 millisecond time bin. And we're graphing it, we're color coding it according to the intensity. But then we can also get, so we can get 2 graphs. The other one is color coded according to the lifetime. And so that's the key thing, is we can get this correlation between the intensity and the lifetime. Another thing that we're very happy about is that our localization precision is down around 5 nanometers. Okay. And that's about as good as you can get with super resolution. In fact, we have actually gotten even better than that recently. We're down to around 1 or 2 nanometers in some of our more recent work. Okay, so now here is the beautiful data that lets us know that we're able to do what we want to do. So now, again, we have the intensity trace. We're seeing the quantum dots blink on and off, and we're seeing that sometimes there's multiple quantum dots and sometimes there's just one quantum dot. We have our lifetime, and you can see the change in the lifetime that is happening along with these changes in intensity. And then we're seeing the change in the X position that is happening along with these changes in lifetime and in intensity. So what's happening is these quantum dots are blinking on and off. And when one quantum dot blinks off, the position that we measure shifts to another quantum dot. Okay. And so we're as we build up this data, we're starting to build up an image that's showing us where the different quantum dots are located. Here is the super resolved image that shows color coding according to intensity. Here is the super resolved image that shows the color coding according to lifetime. You can see that the higher intensity corresponds with the longer lifetime. And you can see that the lower intensity corresponds with the shorter lifetime, that's exactly what we expect for energy transfer. Energy transfer happens from short lifetime donor to long lifetime acceptor, low intensity donor, high intensity acceptor. There's really no other explanation for what's going on in this data. And so that kind of confirms our idea that we're seeing this energy transfer process. And then this is a nice picture that we made for the cover of the journal, where we published this in Applied Physics Letters last year. And we made this nice picture that shows the lifetime on the z-axis, and then we have the X and Y position, and then we're color coding it according to intensity. All right. I think that is where I would like to stop. Well, I just want to before I stop, I want to point out that that Jen and I actually just sent out a big proposal that we're going to try to do this on silver nanoclusters. So we want to, I've had this idea for a long time that quantum dots are not the only interesting type of particle. And silver nanoclusters are also very interesting. And so, we want to try to apply these ideas to other types of systems like silver nanoclusters and other things as well. So with that, I will say thank you very much once again for having me and for letting me share this with you. And I would be happy to answer any questions that you may have.
Absolutely fantastic. I see a ton of hands already. I'll let Gabe start, and then Dallin and Castaneda and Jorge, and then Baudry.
Yeah, thanks. Alan, phenomenal talk. I sent you a message. We'll have to talk later about some ordering stuff. But because I do have a question in regards to the ability to control distance, and have you seen impacts like so on this one, for example, can you correlate that to distances? Are we talking ligand-ligand interaction? Are they, can you tell if they're actually intertwined, and what the limitations are to getting this type of energy transfer process opposed to just individual blinking?
Yeah, those are really good questions. So what I can tell you from this data that I'm showing you here is that the distance between so if you just kind of imagine that this group of dots is one particle, and then that group of dots is another particle, the distance between them is about 12 nanometers. The quantum dots that we're looking at here are they're these CAD selenide, CAD sulfide quantum dots that have a very thick shell, and they're about 10 nanometers in diameter. And so just based on this data alone, you would think that maybe the ligands maybe they're separated a little bit by ligands, but not by very much. Based on the electron microscope image, we don't really see ligands in the electron microscope images. And we don't have electron microscope images for this data. But for the other data that I showed, so those are questions that we have that we have not answered. What is the importance of the ligands? Do they, do the ligands actually move out of the way when they combine together, or do they intertwine with each other? We don't know. We're not sure.
Okay, great. Thanks. Yeah, I'll follow up with you in an email. We've been working with Jen Hollingsworth on probably the same dots, and I'll send you an email because we've seen some interesting ability to order these things.
Oh, okay.
So this could be really interesting, but I'll follow up with you. Phenomenal talk. Thanks.
Yeah, thank you.
Okay, Dallin.
Hi, thanks for the talk. And I had a question kind of on these acceptors versus donors, and like what governs which of these molecules are the acceptors and which are the donors?
Yeah, that's a really good question. So we're working with monodisperse quantum dots, which means that they nominally all have the same size, and they all should have the same, and the size, the thing that governs the band gap energy. Let me go back to this. Let me go back to this picture here. So the band gap energy is governed by the size of the particle. As the particles get smaller, the band gap gets wider. as they get larger, the band gap gets smaller. And so what happens is that even in these monodispersed samples, there is a distribution of sizes, and that's going to give us a distribution of these band gap energies. And so what happens is that when we cluster them together, there's always going to be one that has the smaller band gap, and the other one has larger band gap, and the one that has, and the energy is going to flow from the smaller band gap to the larger band gap. And so, we could maybe speculate on, if I go back to this picture that shows the electron microscope image, we could maybe speculate that this one is bigger, and so it must have the larger band gap, and so it must have the smaller band gap, and so it's the acceptor. But it's hard to do because it has a shell around it, and we don't know what part of it is the shell, what part of it is the core. And so those are another good question that we don't quite have the answer to.
Castañeda?
Thank you.
Yeah, so my question was regarding your new setup for your imaging technique and the importance of those 4 pixels. And as far as what exactly is that field of view that those 4 pixels are able to pick up, and the intensity readings that you were showing, are those intensity readings for a wide range or a wide view of those quantum dots, or is it only a specific spot, like an aggregate of those quantum dots as you were showing?
Yeah, so an aggregate of quantum dots is going to, because they're very close together, if they're all giving light off at the same time, then they're all going to then we're just going to see one blob of light. So basically what we're doing is we're using these 4 pixels to collect this big blob of light. All right. I think of it as a blob of light. And so the idea is that, and the size of this blob. So if you think about the size of a point spread function, it's going to be something like, it's going to be a few hundred nanometers in size, and then we're magnifying that by a factor of 100, and so we're going to essentially be pretty close to 100 microns is going to be the size of this blob of light, and then what we're doing is we're monitoring the intensity of each pixel. And so if this group of particles is shifted over to the right just a little bit, then there's going to be more light on the right-hand side pixels than there are on the left-hand side pixels. And so so basically how much light we collect on each one of these pixels is going to tell us where the little particles are located. Okay. And as they blink on and off, we see that shift a little bit. And so that's what we're measuring. Does that make sense?
No, that makes sense. Thank you.
Yeah.
All right, Jorge.
Hello, my name is Jorge. Thank you so much for your time with us.
Thank you. Thanks for having me.
I'm a graduate student. Thank you. I'm a graduate student under Gabe Montaño, and I'm learning about AFM. Mm-hmm. And I have a 2-part question. I remember you saying, well, is it true that you said that you were using TEM to try to achieve higher resolution because AFM didn't work?
Well, so our particular AFM, I mean, it depends on which AFM you're using. We bought our AFM back in 1999, and it's over 20 years old now. And so our AFM does not give us the resolution to be able to see. So let's say that the quantum dot cluster looks like that. Our AFM image looks like that.
Right.
Okay. And so it's why we started trying to use electron microscopy, because the electron microscopy looks like that. We can see the actual individual particles in the image. Now, so there are AFM techniques that are better than the ones we're using that probably could get that level of resolution.
So that was my second question, is why you went that way and not try to go low temperature, low vacuum with AFM.
Just because we're trying to do things as easy as we can. You know, I think that that is another way that we could go, but I think we're trying to do things. We're trying to do things as easy as we can. Make it as easy as on ourselves as possible.
Okay, thank you very much.
Yeah.
All right, Ryan.
Professor Van Orden, thank you so much for
You're welcome.
a wonderful talk. That was fantastic.
Thank you. You're welcome. You're welcome.
Actually, this slide right here goes right to what I wanted to ask you about. So I was super intrigued about these lifetime measurements that you were taking, and when you're comparing a single quantum dot to clusters. Mm-hmm. And I was curious, do you see any evidence for superradiance?
We do not. I'm not sure exactly what we would see if that was happening.
So you should see decay rates increase with the number of particles, and intensity increases with the square of the number of particles.
We have not seen that, but it may just simply be that we haven't looked for that. Maybe it's something we need to look for. You know, because that would be very interesting if that was happening.
Thank you.
Yeah.
Alan, I have a quick question too. So in the 2020 article, just looking at a single quantum dot, you had this decently sort of averaged intensity over the surface of the QD. Mm-hmm. Whereas in like the JPCC 2018 Doug Shepard era paper, you're looking at a single quantum dot, you almost saw this like bimodal distribution of intensity, where the highest intensity was sort of on one side, and the middle intensity was on another. Is this a way for us to kind of tell something about the QD structure, anisotropy, or I don't know, layer issues?
I am not sure why this looks like this. I asked myself that same question, is that I wonder if maybe we're seeing, I mean, for one thing, you know, the red is lower intensity, and so it is going to have a broader distribution, but why isn't the green centered on the red is the question. I'm not exactly sure why that is, but I did have the same question that you did. We do see other images that aren't, I mean, they're all kind of different, you know, everyone is a little bit different. And so this is just what it looks like for that one particular particle.
And while you're on this slide, what do you use as fiducials going back and forth between like super res and electron microscopy?
Yeah, we just kind of measure it relative to we have this little teeny grid. And whenever we're doing our measurements, we go and we measure the corner. Where did I see that picture? That picture is here. So we have this little grid. And so you're going to have some particles on this part of the grid, and we just measure the corner, and we measure where the particle is relative to that little corner of that little piece there. And then we go and we give it to Chris, and we say, go find the particles. Here are the coordinates. And it's a hard experiment. It's a hard thing to do.
Any other questions?
I have a question.
Badri.
Yeah. My question is already asked by Gabe, so thank you for that. I will follow up with that one. So you said the distance between these quantum dots was around 10, 12 nanometer, right?
Yeah, that's what we think.
So when you do the same experiment with clusters, it would be in the scale of 1 or 2 nanometer, the distance between them. And then what would be the photon transfer sort of mechanism? Would it be through the ligand or?
Well, yeah, I mean, we're not really sure what the mechanism is. The one possible mechanism is a resonance energy transfer type mechanism. And that's basically just an interaction between the dipole. It's a dipole-dipole interaction that happens through space, doesn't depend on ligands, but like I say, there are other possible mechanisms that could also give the same answer. And so we're kind of agnostic about what is the exact mechanism. We tend to talk in terms of resonance energy transfer, but that may not be what it is.
And then do you have any sense of how far apart these quantum dots needs to be to behave individually compared to behaving as a cluster?
Yeah, so a typical Forster radius. So the way you characterize the resonance energy transfer efficiency is with this quantity called the Forster radius, and that's the distance at which the energy transfer efficiency is 50%. And for these quantum dots, it's on the order of a few nanometers out to about 10 nanometers. And so if you have two quantum dots that are 10 nanometers apart, you could have 50% or more efficiency.
And on that note, I mean, where do you sort of fall on? I remember, how does that usually scale? Inverse to the sixth power?
It's 1 over r to the sixth.
Yeah, so I remember Jeff Strauss came out saying that he thought that QD energy transfer was more of 1 over r to the fourth.
Yeah. And
where do you fall on that?
I don't know.
You don't want to say anything on a recorded lecture?
It could be 1 over r to the fourth. It could be 1 over r to the sixth.
Okay.
I think when people are talking about, a lot of people measure energy transfer between a quantum dot and an organic dye molecule. And I think when they do that, they're thinking 1 over r to the sixth. But if it's between quantum dot energy transfer, then I'm not sure. I think all bets are off at that point.
Okay. Any other final questions?
John, I have another question. So I think you talked about earlier that there are clusters that don't exhibit the blinking and the change in lifetimes.
Right. Yeah.
And my question is, so are those like, do you consider those clusters or just those particles just existing close to each other and not interacting?
Yeah.
Or is it some difference where there's no acceptor and donor type of relationship?
It could be all of the above.
Okay.
Yeah, these are the questions that we're trying to address, that we're kind of driving towards.
Alan, as always, just a fantastic talk that got so many people thinking about how to use this for their own capabilities, challenging their own ideas on their work as well. It was fantastic. We really thank you again for chatting with us.
Thank you, and thank you for having me. It's been fun.
All right. Thank you so much. Appreciate it.
Thank you.
Thank you.
By the way, Professor Van Orden, I postdoc